Okay, very good. All of you will talk, I will not. Thank you. Thank you for coming back after tea. You had a very interesting and high-class performance from all the three of them. Some of them had to do it twice. I must say, from my vantage point of view, a lot of it was over my head. I didn't quite understand what was going on. But all I know is it costs a lot of money to do all these things. And for Singapore's prominence in the healthcare system, we cannot do without it. As new science, new technologies come on board, somehow we need to provide them for our patients and for the public at large. So there's one question that is put up for the panel to discuss. Maybe I give each of them about 3 minutes to say something about this topic, and then we'll move on to the Q&A, because I think many of you have questions that you would like to ask. So the discussion is on this topic: cell, tissue, and gene therapy products. Are they as good as they claim? So after we're done with this, we'll do the Q&A. So anybody can start. It's a democratic process. Ladies first? Yes, please.
I think the short answer is we don't know. I think a lot of these products are still very early in their use in clinical practice, and we haven't had a lot of follow-up of patients in clinical practice with the use of these very new products like the CAR-T therapies. We also know that the use of the products have primarily been in those, most in need, who are generally smaller populations of people. So I think our information is that the jury is probably still out. They certainly work. How well they work, I think, is where the uncertainty is at the moment.
But what are the parameters you're looking for to say that they will be good? How long would you take the time frame to calculate the value or the costs?
I think in diseases that are very rare, you need follow-up of those small groups of patients over a period of 5 or more years. That's the usual projected time for time horizons in our sort of assessment. For diseases which are much more common, and I use the example of multiple myeloma, you would really need to look at randomised studies around the place of these treatments in relation to so many other new therapies that are coming to the fore. So this might be the precursor of therapies that could be a lot better. And so time spent on long-term evaluation of some of these sort of initial therapies is already being diverted now to looking at even better treatments. And I think that's really a way to think about it for more common cancers, for instance.
Yeah, I guess the question from an HTA perspective is, are they good enough? Because whether they work at all is a question you might think is more for a regulator. So for the medicines regulator in the United Kingdom, if a drug doesn't work or is unsafe, it's for them to intervene. I think we're in the somehow invidious position to have to decide whether something is good enough and whether the risk we're taking by displacing health from others, by spending a lot of money on a drug like that, whether that's okay. By saying that, I also think we have almost a moral responsibility to make sure we understand whether the drug works and not just whether it's okay in cost-effectiveness terms. So we are keen, as I've put out in my talk earlier, we're keen to follow these drugs up, and we do that through the Cancer Drugs Fund in particular now, but also the Innovative Medicines Fund. So there's an emphasis on trying to find out whether they're good enough, but a real interest also in understanding whether the modeling we're using is indeed representative of the effect of these technologies. So there's, of course, a big hype, because in the United Kingdom, about one out of 3 companies that are interested in ATMPs are based in the United Kingdom. 12% of the ATMP trials are done in the United Kingdom, and that's a 20% increase year on year. So there's a real interest, and to push against that, I think, is the challenge we have as a HTA agency.
But for the one that treats the gene defect for blindness, does it work? Yes.
Which one?
For the blindness, Laksana.
Yeah, well, the jury is out, isn't it? At the moment, we think it is worth spending the extra money under a framework that we have established for highly specialized technologies. But I think it is up to us as a society, academics, regulators, and HTA agencies together to understand whether the signal that we're using in the modeling is indeed correct. But that's difficult because the moment you've let the technology onto the market, the incentives to continue to collect the data may not be there. And it also depends what kind of data you use. Is it real-world evidence? Is it continuation of the clinical trials? And I think this issue becomes ever more important as regulators move earlier into the development program by providing signals for use. It makes the job of a HTA agency more difficult.
So for single gene diseases which cause obvious defects, it's best to treat in utero. And maybe Han Chong will give us some figures of what they think are the common single dominant gene diseases in Singapore. For example, if blindness due to this gene, how many cases are there? And therefore, what should be the cost? Should it be calculated, annualized, or throughout a lifetime, or just the cost of the drug, just once? Because it is a cure, right? It's touted as a cure, not a chronic disease.
So for the hereditary retinal dystrophies, actually, I'm not Ang Tin or Wong Tian Yan, but I don't know the numbers, but I think if you have no option and the person's going to go blind, versus getting Luxturna, right? Getting Luxturna and paying over $1 million and perhaps restoring some sight and being able to recognize people and loved ones, it's definitely something is better than nothing. But as correctly said, it comes at a price. So is it good enough as a cost-effective drug? I think if it's $100, the answer is yes. But if it's more than $1 million, which it is now, and I'm not a regulator, then it's a difficult question to answer because the series are small, the long-term outcomes are not known. It's the same with spinal muscular atrophy. If you're a child, you can't walk, and then suddenly you get this gene therapy for $2 million, and then you can start to walk. And in small batches, you begin to see people improving. Of course, something is better than nothing. But again, it's really about the price tag, and the cost of goods hopefully can come down. But beyond that, I have absolutely no experience with either diseases. So, yeah.
So can you comment from your perspective of the Singapore scene? What are our challenges regarding these two therapies?
Well, this sounds like a grand round with Prof. Chi.
It's okay, it's back to
when
the
I was
good
a
old days.
resident. So can I comment? So I guess because I was very impressed that NICE was willing to reimburse not just CAR T cells but Luxturna. To me, that was remarkable because it is a very expensive drug, and I don't know what negotiations take place between NICE and the drug companies. Of course, the other rare condition, and Prof. Chi talks about this, about rare Asian condition, is beta thalassemia. And as you know, that's even more expensive. I think that's $2.8 million per injection to treat beta thalassemia. And will that be cost-effective compared to regular blood transfusion? I don't know either.
I guess another way of thinking about it is that when I showed the slide on Luxturna, the QALYs, that's the benefits we calculate, attributed to that drug over a lifetime were about 10 on average for a patient. Now that might not sound a lot, but if you think about bog standard drugs, maybe 0.5 of a QALY or even less. So the ethical question is more, do you want a lot of benefits for very few people? Or do you want a lot of benefit or small benefits for many people? Now that's a difficult question. We kind of equipoise at the moment. We calculate just the value of Luxturna compared with doing nothing or doing something else that might be relevant in that disease area. But don't compare rare diseases versus non-rare diseases, because that will be a completely different debate.
Okay, I'm waiting for questions from the floor. How long do I have to wait?
Actually, on the area of CAR T cells, we have done some health cost-effectiveness studies, and Prof. William Huang is actually in the audience, and he actually treats patients with leukemia and lymphoma. So I'm not sure whether, William, would you like to say a few words about those studies?
Okay, so we move on to CAR T cells. So you remember the motor car running around the brown field. So William has got experience and does it for patients as he has published. Can you tell us what are the challenges and what do you want ACE to do?
Han Chong, I'll talk to you after this.
It's okay, it's only good things.
Well, anyway, if we are asking the question of whether cell and gene therapies are worth it, I find it's a difficult question. It's like asking, are tablet medications worth it? Because cell and gene therapies is just a form of treatment, right? And there are many different forms. And the answer to that is really very specific to the kind of treatment and the data for that particular drug that is being, evaluated. So whether it's even CAR T cells, there are many different kinds of CAR T cells. One may be worth it, and one may not. Gene therapies, one may be worth it, and one may not. So need very specific answer to that. The second aspect about cell and gene therapies is that the time horizon needs to be very different in the evaluation, because most of the drugs we take, an oral drug will last a few hours, a few days at most. An intravenous drug, even a monoclonal antibody, lasts a few weeks, and then it disappears from the system. Cell and gene therapies go in, and many of them can last for the lifetime of the individual, and they continue to have a therapeutic effect for a long time. So much so that if you give a CAR T cell therapy, it can continue to be there. Even in Emily Whitehead and the rest, they've detected the presence of these CAR T cells 10 and more years after the original infusion. So in my own experience with patients receiving CAR T cells, as well as other therapies, although the cost-effectiveness comparisons have been generally against one type of therapy compared to CAR T cells, for example. But however, if a patient has a relapse, that patient doesn't, nowadays with all the therapies we have, doesn't necessarily, succumb to the illness because there will be second-line therapy, third-line therapy, fourth-line therapy, and many therapies that can continue to prolong the life of the individual, although it gets more and more complicated with time. And many of these therapies can cost $20,000 a month sometimes, and we project it over a course of 2 years. The cost of that is really considerable. So when we look at therapies for cell and gene therapies, whether CAR T cell or otherwise, that is certainly something that needs to be considered. Most recently, one of the patients I have went to see her GP, and the GP was just so happy that she's free of lymphoma, free of leukemia, free of lymphoma, and free of needing any complicated medicine at the point of time. And the GP shared that she's so happy because her own daughter is working with us in NCC. So so that's just on a personal basis.
But just to push it further, in your patients, anonymous, what was the payment or financing for the treatment?
Okay. So this one was on clinical trial, so it was free.
If not, how?
So yes, it is a problem. Some patients do have insurance that will help, together with some of their own finances. We got our medical social worker. We also run clinical trials, and the main reason we run clinical trials is really to get the patient, free drug actually. But of course, on the long term, we have to think about how this continues. I think at the end of the day, the ability to pay needs to have everyone contribute. It needs to have the hospitals need to contribute in some way to reduce the cost for the patient. The insurers need to contribute. Government needs to contribute. The drug company needs to be able to bring the price down. Everyone needs to meet somewhere in the middle to benefit the patients as best as possible, because the greater the numbers are for the patients, the greater cost effectiveness you get in the system, the greater efficiency we get in hospitals as well as at the pharma side as well. So that's just my own small little perspective.
Yeah, so we have to think as a society, as a nation, who pays, you see. If you say government pays, means taxpayer pay. Taxpayer means you and I pay. All right, so it's as simple as that. So are you willing to foot so many $1 million or X amount to treat one patient or 10 patients or 50 patients? Is the answer yes or no? So you need some evidence to say, what am I buying? What is the value? Right? Of course, if it's your own relative, someone related to you, your answer probably will be yes. But for society at large, it's a bigger question, and there are other aspects to be considered. But for the UK and Australia, probably when they are state-funded, or majority of them are state-funded, plus insurance, the consideration will be a bit maybe simpler. You just say, okay, this is the budget for you. As many patients as you treat, you just treat. And if the money runs out, you come and ask me again. If the minister is changed, maybe you have better luck, I give you some more money. If the old minister is still there, said, I told you that's all, then that's all. Wait for next year. So for us, we are a multi-payer system, so we need to think as a society, how much can we afford? Therefore, the basic question for this panel was, is this real? Do these drugs actually work? If you treat the gene, you correct the gene defect, are they cured? Meaning for the rest of their life, you don't have to bother about them. So we don't have quite the answers yet. But it's a good thing that having approved these drugs, they are asking for data to track and see what exactly they have approved, what exactly has been the value. So we don't want to be the first runner in these kind of advances. We want to see and learn from other people. So my question to you is, can we learn from you? What other drugs have you approved which after 5 or 10 years, you have said, yeah, this is value for money, and therefore we should be able to say, we should also agree based on our contextual, cultural considerations. Do you have such drugs? To Han Chong has.
No, so in terms of actual numbers for CAR T cells, I think just among Singaporeans, we have about 5 or so relapsed ALL that we need to look after per year. And we have about 45 large cell lymphomas refractory that we need to look after. So it's kind of having to decide whether you want to put the budget out for 50 people a year. I mean, for the Indonesians and the Malaysians and the Vietnamese who come to Singapore, unfortunately, as you know, there's no reimbursement for those guys. So those are the actual numbers for CAR T cells.
Yeah, but for your CAR T patients, are they curative?
I think William is best answered, but I think 80 to 90% of patients will go into some form of profound and significant remission. And of those, I would say 50 to 60% will still remain in remission for a very long time. And as William has already alluded to, these drugs can stay in the body. So that's a Nature paper by Carl June. Inside the body, they're still there for 10 years. Whereas if you give chemotherapy, within weeks, they are passed out in the urine or something.
Yeah, Robin?
I was just going to say on the ALL CAR T example, that's a really interesting one because effectively that CAR T was positioned for, as you've just said, for children who have failed every other treatment, and it's sort of a rule of rescue. And in Australia, we estimated that there would be 23 children who would be actually eligible. And so for us, that was sort of really, that seems like a very reasonable thing. But interestingly, in reality, the number of children treated have been far less than that, probably around half that number. And I think that sort of says that the sort of fitness to have the treatment was not necessarily quite there, even though the case was made that these 23 children would be all ready and able to be treated. We sort of overestimated whether these people would really be able to be rescued because they weren't fit enough otherwise. So I think that's the same has held true for diffuse large B-cell lymphoma, mantle cell lymphoma, also reimbursed in Australia, is that we've tended to overestimate the people, the population who truly you could rescue. And when it comes to the clinician wanting to treat the patient, there's actually less people that are actually eligible for the treatment than we initially anticipated. That's happened almost with, it has happened with every CAR T treatment so far.
Do we also see them dropping out earlier through the research? Because some data that we've seen is that there's a lot of high-cost technologies coming. A great percentage of those are ATMPs, but actually when it comes to licensing, a lot of them drop out before they ever get there. So you know, there might be an argument that the ones that do make it have been scrutinized significantly. And maybe the question of whether they will work is one that we have answered earlier already. That may be different than another tablet or another IV fluid. So I wonder whether that's one of the angles. I do think, though, when we as a society invest in that kind of technology, we do have an obligation to continue to collect data. And we do that through those medicines funds that are separate from the general reimbursement. I think that's a really good model. But it shouldn't then make companies think, well, I have to do even less preparatory work because it will all be tracked once we're in the market. That wouldn't be right either.
Can I just ask for gene therapy? You mentioned the earlier you give it, the better, before any damage from a disease has occurred. Would it be possible to give it in utero?
To be
Given in utero,
Oh,
before
in
the
utero.
baby is born.
Well, probably not. That's probably, at the moment anyway, that's certainly, right out there. But I think in terms of, as I said, screening the population to identify people who are carrying recessive genes, so biallelic recessive genes, so both genes are abnormal, so they're going to manifest the condition in some form, and knowing that if the person and the couple are proceeding with those pregnancies, that as soon as that child is born, that you can get started on treatment is a paradigm that's being pushed, because obviously in the retinal disease example, if the retinal cells have already deteriorated, you can't fix cells that are already died. But if you need enough tissue reserves to actually make gene therapy work. So if the cells are dead, there's nothing to work on. So treating people before they've lost their vision, in the case of the children with Lux Turner, you really do need to treat them early before they're blind. Once they're blind, there's no restoration of vision, is very low. So that's identify early, treat early, you'll try and get maximum results, but that requires the health system to do a whole lot of other things like screen for these conditions and, and get into some of the ethical issues of, is it reasonable to screen for these conditions? What decisions will families make, will parents make in relation to finding out that their child has these conditions?
So I think in utero is very sensitive, because if you treat a child before they're born, I think, as Professor Ward was saying, you don't know what's going to happen in terms of off-target effects. Gene editing is going to come as the next wave of therapeutics, and there was a Chinese investigator who actually treated in utero by adding a gene to prevent HIV, because her mom was HIV positive, if I'm not wrong, and that created a huge outroar in the gene editing world, and I think that became a really big problem. And I think the Chinese government censored him as well, because even though he had the best of intentions, I think the answer is we don't know how the gene editing is going to affect that future baby in other ways, and I think there's a lot of regulations about these things right now.
Okay, so your old patient who's running more than you can walk in a month.
Yeah.
How is he doing, and who is still paying for him?
So thanks, well, pharma was very generous. So he was on this trial for 2 years, and I think beyond 2 years, they said, well, you can stop the drug. The truth is nobody knows, and he's done really, really well. His tumor is super small, and so Roche, the pharma company, said, well, we'll give you compassionate use for this drug, and you just pay a really tiny amount, and he's paying a very tiny amount. And so they continue to give him these drugs that normally cost $15,000 a month, FOC, almost FOC. So he's obviously a happy guy, and of course he made a donation to the National Cancer Center, so we're very happy as well. And William is very happy. So I think this, but again, I need to be very clear that only 30% of patients respond to these two drugs with liver cancer. And to be very clear, maybe another 30% have stable disease. The median survival is still 19 months only, so it's by no means a cure. He's very, very fortunate as an individual.
Back to value-based healthcare. On behalf of ACE, do you all define value differently for different modalities or medical devices? And how long is the patent for the new technologies? How long does the patent last?
We don't value health benefits different according to the modality. So it wouldn't be, if you get a medical device, you're worth less than a medicine or something. That would be rather strange. No, it's about unmet need is one. It's about unmet need and rarity. That's what we have a citizens' council or a group of people from the public that we ask some ethical questions, and they tend to come back with answers that are about severity and unmet need when there's any differentiation between what you might consider to be more valuable. So we have for a long time had an extra modifier benefit for end of life. So where normally our threshold is, say, £30,000 per quality adjusted life year, when you're at the end of life, it would be £50,000 per quality adjusted life year. And when you are suffering from a ultra-rare disease with enormous severity, and Luxturna fell in that category, then we would go up to 100 to 300,000 pounds per QALY. Now that is a choice we make as a society, and we can make that choice because we have one payer, we have a tax-based healthcare system. So a lot of the questions you ask are not that relevant for us. But equally, within that system, you would still need to manage risk properly, because this is all based on significant assumptions about health benefits. And I think this is where perhaps the story needs to go, is that it's staring at whether those drugs are really effective. That's important, but are we managing the risk of them not being effective and displacing health for others? Are we managing that risk properly? And are our payers engaged in that conversation? I think is the critical part.
No, I absolutely agree with that. But I guess the only other exception to that is certainly for a therapeutic intervention, we definitely, it doesn't matter whether it's a stent or a drug, it's the same deal. It's, quality of life, life years gained. But in the diagnostic area, we've increasingly attached value to what we've called the value of knowing and the value of naming. And in a sense that by knowing something, our society has told us that they feel that, now this doesn't get, $500,000, for knowing something, but people have being able to name a disease, to call it something, so that even if there isn't a treatment, our society in Australia at least has said that they attach value to being able to name that condition. It allows them to join support groups, to have a better engagement with disease, understanding its natural history. So we've introduced this term in our guidelines in the diagnostic sector called value of knowing and value of naming, because effectively Australian government will pay, and is paying now, dollars for that information. It's nothing like what they would pay for a therapeutic intervention, but by the same token, we've recognised that society places a value on that, and that has a monetary value that we have now attached to that information.
Can I ask Professor Watt, in your slides you showed that growth rate of expenditure on biotech is going at 11%. How do you see the future of this, and how would you manage?
I think that is continuing to grow. I mean, the slope of that curve is actually getting steeper, not shallower. So I think that is going to keep growing, and we're going to see more and more tissue-engineered products, regenerative medicine products. And because a lot of them are so personalised and so labour-intense to produce and bespoke, we're going to see, I think, a continued rising of those products. That's where the most interest is, at least in the analysis that I did for this, for looking at industry.
The way to manage that is to bring down costs?
Is to bring down costs, or in the unit cost of the product, but also I think there's this other issue of our supply chain and how expensive it is to actually even implement some of these things in the health system. We know that for some of our products, like the CAR-T treatment, it costs us about the same as the product cost for actually the health system to implement it, because it's a lot of salaries of staff and a lot of manpower. So we're looking at how do we actually make that more efficient as well, because if we're paying twice the cost every time we need to do this, we need to be able to be more efficient and more adaptive in the health system to be able to even absorb these products.
And it is not just, the cost of those individual products. It's also used in combination, used in sequence. You see it in spinal muscular atrophy, that there's nusinersen and then Zolgensma. It builds up, and if you use a relatively strict and rigid value system, then you might end up paying a lot more for the health benefits overall than you might have by having all these combinations and sequences than you might have had if you had one shot. I think there's a lot of further economics to be done on this, I think, more than the clinical effectiveness signals.
Right, my big clock here tells me I have 10 minutes left. We talk a lot about economists and the high-powered economists. Are there any around? Would you like to say something or tell us how to unlock this conundrum? Or have they all left? Tuan Chong, any of your friends around? Where's Jeremy? No, Jeremy not here. Wei Yang also gone. Tuan Chong, your friends have deserted you. Do your government set a value on how much you can spend on these kinds of technology?
So our government hasn't capped the expenditure on this or on pharmaceuticals in general. We have uncapped, essentially, budgets for both.
Yeah, and as I pointed out, in the UK, there is a voluntary scheme where pharmaceutical industry has agreed with the government that the total sales of pharmaceuticals cannot go above 2% a year. And there, within that scheme, it's very complicated. There's incentives for innovation. So if you are the manufacturer for cell and gene therapy, in the first 3 years, the sales for that innovation are excluded from the payment you have to make. Conversely, it therefore means for those companies that are not innovating, they are there, in fact, paying for the innovation like cell and gene therapy. So it's a complicated matter. It pushes some of the risk back on the industry, which I think is good. But to have a pot, there was some conversation about a pot for ATMPs or cell and gene therapies, but that, I think, as you said before, that doesn't really make much sense because it's just a modality.
Can you expand for us what is iLab?
Yeah, well, iLab, the Innovative Licensing and Access Pathway, tries to ensure that when regulators think about, for example, cell and gene therapies in a certain way, about the effect size they request and they expect for technologies to come to the market, we see that regulators do that earlier and earlier. They take bigger risks because it's a competitive environment in which these medicines are launched, especially now between the UK and Europe. And we see as HTA agency that there's a gap between what a regulator accepts as evidence and what we accept as evidence. And we know that we've known that for many years, that the endpoints we require are not the endpoints necessarily that regulators look for. So what we're trying to achieve in the Innovative Licensing and Access Pathway is to find those new modalities that Professor Ward so clearly described in her second talk, and try and come up with plans for how we deal with the evidence we require, the core outcomes we jointly think are relevant, and qualify and validate those up front so that we don't end up at market entry with an emotional picture where patients and carers say, well, there is this new gene therapy, the regulator has said yes, why is NICE not jumping to it? And I think the only way to answer that question is to work together early and get the evidence right.
If it's no secret, can you tell us in Australia, what are the 3 latest either drugs or technologies you have been approving for this year?
Well, it's in the public domain now. The CAR T treatments for mantle cell lymphoma, which I think you had is still to be determined, that's been recommended. The diffuse large B-cell lymphoma, ALL, a while ago. So all of those CAR Ts have been approved. Multiple myeloma is one that we've seen that hasn't been approved. So I think that is the beginning of looking at when you're expanding out the treatment to much broader populations of people who have got multiple other modalities of treatment available to them. So they've all been approved.
What about medtech?
Medtech. Our biggest things in medtech are largely around cardiac devices. So, insertion of stents to relieve, to remove clots during TAVI procedures. Lots of the interventions, the cardiovascular interventions, we're seeing a lot more of those. Really started with TAVI, but now is many more of those cardiac interventions preventing stroke, etc.
What about in the UK, the latest approved?
Well, there's a great emphasis on digital technologies. So the Healthy IO is a company that makes a digital device. We've just approved a heart monitor, a digital heart monitor, and I think there again the question for us is with medtech is are there ways of allowing these products on the market while evidence is collected? There's always been an assumption that medtech has very little evidence in the first place. Well, that's almost at parity now with pharmaceuticals. For both, we need to have systems in place to track evidence, and so for those technologies, we're looking at early value assessment plus continuous tracking.
Okay, I have less than 5 minutes. Any more questions from the audience? Yes, please, Bing Sing. Ask a money question. He's a money man.
Yes,
so-
I'll ask a money question. Bing Sing, MOH, and in the spirit of Prof. Chi saying that Singapore likes to learn from other countries, I've got a question to learn from managed care, managed access, and so questions from Mr. Boyson. My question is, how do you disinvest once you start? So it's easy to start. It's usually very challenging to stop. So could you share your experience in stopping something you have started? How do you manage that process? And my second question, which is related, is how long is managed access for? Because for some of these new treatments, the time horizon you need to evaluate that they truly work is pretty long. So how do you decide when to stop? Thanks.
Yeah, that's a really that's a really good one. We haven't had that many experiences yet of things that come out of these funds, the Cancer Drugs Fund in particular, where we have established they're not cost effective. We had an earlier version of the Cancer Drugs Fund where it didn't really matter what the price was because we were not involved. And when we looked at 20 drugs that were in that fund, effectively at their list price, when we looked at those drugs 2 years later, companies were more than willing to make a deal. So there's a dynamic about pricing at market entry and after a couple of years. And often we see companies being able to then come to the table. So actual disinvestment, as in, let's say, no altogether, happens very rarely. We might optimize, so we might say it goes only for these patients with these eligibility criteria, plus the company coming down on price. We originally said that the Cancer Drugs Fund was for about 2 years maximum per drug, but of course we see now with the ATMPs, cell and gene therapies, you have to extend that. So we see 5 years as the maximum at the moment.
Well, I think absolute disinvestment we've had no luck with that. We had a Senate inquiry into one thing that we tried to disinvest in, and I think that was the end of the attempts to try and disinvest. But I think there are ways in which, pressure is brought to bear. Either the thing doesn't work, which is the simplest form of disinvestment because doctors stop using it, so it falls off anyway and you don't need to worry about that. But where the marketplace is hungry, and I mean the clinicians are prescribing it, then pushing more products into that same financial cap, is a form of disinvestment because it squeezes the amount of money that's available for that whole class of therapies. So it's a form of reducing the the price, but you can't disinvest entirely and say the product is no longer available. But you can actually, do things like forcing them into groups, which then share the same cost cap, and that's a form of disinvesting in terms of how much we pay for a given product. Those are the sort of only forms of disinvestment, if that's what you want to call them, but it's financial constraint. It's constraining the budget expenditure is really what we're trying to do there.
Where there's opportunity, sorry, I forgot to say that, is through our clinical guidelines program. So in clinical guidelines, there's there's many opportunities for identification of products that are no longer in clinical view the right option. And when we have gathered that information and the payer in the NHS use that to then say to clinicians, this is, we know this doesn't work. Your peers in the clinical guidelines have established, do not do this. Then that's a much better stick than in the kind of highly innovative pharmaceutical world to enter there in disinvestment. That's a much harder sell.
Right, so let the patients know that this drug you're taking doesn't work and tell the doctor to stop it. Like now you go for head injury, they said, do skull x-ray. No, they do CT scan. So you want to order skull x-ray, nobody knows what it's all about. Because no point, they just want to know whether you're bleeding or not. So it's all CT scan now, rather than skull x-ray. So very hard to find a skull x-ray. Okay, my time is up. I don't know about yours. So thank you for staying. We enjoyed talking with you and talking among ourselves. Glad you tuned in, and I hope you got some gems from what you have heard from our distinguished panel of speakers and experts. I'm not the expert, so I wish that you will continue to discuss with them your problems. I'm sure they have solutions. It's whether they want to tell you. Because every drug is cost effective depending on how much it costs, right? So if you bring down the cost low enough, it will be cost effective. So thank you very much. Enjoy the rest of the conference, and maybe just show our appreciation in the normal way.